Welcome to the first video of Biologics. Today we're going to talk about the human immunodeficiency virus, which is HIV.
Let's start with the basics of a virus. How does a virus spread? How do they infect us? Viruses are non-cellular agents that infect cells and reproduce inside them. Each one infects a specific type of host. This includes all life forms: bacteria, archaea, plant cells, and animal cells.
Now, what is HIV in simple terms? HIV is a virus that attacks the body's immune systems. It interferes with the body's ability to fight infections and diseases. It is a retrovirus that converts RNA genome to DNA after infecting the host.
What does an HIV virus look like? The structure contains, first, in the middle it contains RNA and reverse transcriptase, later which is then enclosed by a capsid, and this capsid is then surrounded by a protein called matrix. Then we have the lipid membrane, which has glycoproteins 41 and 120 on it. The genetic material includes a single-stranded RNA genome, which is 2 copies of a single-stranded positive sense RNA molecule. Of 9,749 bases, there are 9 genes coding for 15 viral proteins, including 4 enzymes. The lipid membrane, or the bilayer, this is responsible for protecting the genome to help facilitate entry into the new host cells. The matrix is a protein which is used to describe a protein that forms a layer on the inside of the viral envelope. Matrix proteins play important roles in the virus assembly as they form links or bridges between nucleocapsids, cores, and the envelope.
The next is reverse transcriptase. Reverse transcriptase is an enzyme which HIV uses to convert its RNA into viral DNA once it enters the host cell. Next is the capsid. A virus is made up of genetic material that is encased with a protective protein coat called the capsid. The capsid also helps the virus to infect host cells by binding to the host receptor proteins and releasing its genetic material. Lastly, we have glycoprotein 41 and glycoprotein 120. Glycoprotein 120 determines viral tropism by binding to target cell receptors, while glycoprotein 41 mediates fusion between viral and cellular membranes.
Which cells does the HIV target and when it enters the host? HIV targets the immune system by targeting the T helper cells, particularly the CD4 plus T cells, dendritic cells, and macrophages. When the CD4 plus T cells count eventually declines below a critical point due to action of HIV, the cell-mediated immunity is lost. The body progressively becomes more susceptible to opportunistic infections, resulting in AIDS developing. HIV invades a T cell by a process called the lytic cycle, where first the HIV virus targets the T cells, then the DNA enters the T cell. Next, it copies its DNA and replicates inside the host cell and completely destroys it.
Now we'll talk about how does the HIV virus infect. If someone is detected with HIV virus, it can be transmitted through bodily fluids such as blood, semen, vaginal fluid, anal fluid, and breast milk. People can get HIV through many ways such as sexual intercourse, sharing drug injecting equipments, mother-to-child transmission during pregnancy, and coming into contact with contaminated blood. The HIV virus is a retrovirus that uses reverse transcriptase to convert its single-stranded RNA genome to DNA. Reverse transcriptase, unlike DNA polymerase, does not proofread or correct errors, which leads to many mutations. And that's all you need to know about the basics of HIV. Hope you found this helpful.